So after this introduction, we'd like to start by just quickly saying what context we are with, within what context we are. We are now going to use the knowledge that we have gained to build up models of networks of biological processes. So, and while the network we're going to work on today is very simple, for the case of demonstration, the slide here shows that the theme of negative feedback is one that's widely spread throughout the biological area. And if we think of the example displayed here, that's intracellular signaling, there's not one negative feedback, there's many instances of negative feedback combined with instances of positive feedback. And so knowing how to do one, you'll probably be able to do them all. The important thing is that these feedbacks introduce important regulatory features that we need to understand before we can hope to understand the full network in action. So I'm not going to go into the details displayed here. That's a signaling pathway or a signaling network where there is feedback from the proteins that go into the nucleus to regulate gene expression and then come back and have an impact on the upstream elements of that thing. You can read up about this with the link given. Let's look at how this field started. It's actually a very old story conceptually, going back at least to 1963, or as far as we are concerned today. Brian Goodwin, a theoretical biologist, considered negative feedback in the context of, say, biochemical reaction networks or gene expression networks. And this is what we're going to take as an example where we do very simple conceptual reasoning about what negative feedback in a biological context can do and maybe also what it cannot do. So for the sake of today's exercise, we're going to go to the next slide, and we have simplified this Goodwin model, and we've simplified it such that it's very easy to manipulate and that we have a simple set of equations where we can directly apply all the tools that we have learned so far. So the simplified Goodwin model is schematically displayed at the top of this slide. And what we have here abstractly labeled is two nodes of our network. So it's the smallest node size possible because there's no network with less than two nodes. So we have a very little network of just two nodes connected, and the arrows indicate the kind of interactions that take place with and between the nodes. So if we look at a part of the network highlighted here, the first node called X1 representing, for example, a metabolite or a signaling molecule, or can be some other entity, but in that case, something that is being, in a biochemical context, being produced. So there's an arrow leading into it. Also, there's a reaction that consumes it, so there's an arrow leading away from it. The degradation or consumption arrow leads to a second species called X2, so it's another molecule, another protein, another entity. And this one, apart from being produced with that arrow in the first place, also has a degradation with it, so there's some reaction into something that we don't display with a name here. And the reason we don't specify what is on the right-hand side of this here, because we don't want to talk about it in mathematical terms. Biologically, it means that the arrow into the system and the arrow out of the system help us to realize an open flow environment. Means in a similar spirit as in any biological system, there's an exchange of matter and energy with the environment. Some things can come in and some things can go out. So we'll not end up at zero concentrations because of something being depleted totally, and will not end up with very, very high concentration due to accumulation of any of the molecules taking part in the network. Highlighted in that arrow, arc-shaped arrow at the bottom of the scheme with that red indicator and the negative sign is our feedback, the one that we are going to look particularly into. And of course, the red negative sign indicating negative feedback. And this will then complete a network including feedback. So we actually have an interaction from node x1 to x2, but also an interaction from x2 back to x1. So there's a loop structure, and that's what makes network dynamics interesting. Right, for our purpose, we're going to look at the whole thing, and we have the model equations written down here. So it's simpler than the original Goodwin, which had 3 variables. Here we have only 2 variables. Fairly simple way, one variable labeled x1, the other x2, and we have the model set up in a way that you should have familiarized yourself with. It's a set of ordinary differential equations. So it's a deterministic model, and a model that allows us to make time-dependent predictions, means knowing a state in the present, we can predict the future evolution of it. So I'm inviting Europe to join me again.
Okay.
And after this introduction, in the way for those who are used to it, we're going to come with a traditional entering MATLAB. Taking our webcam away to save some space for transmission. Bringing up MATLAB and starting with just a template where there's only comments to help us explain to you what we're going to do.
Okay, so let's give ourselves some space to start with. So we're just going to focus on this first bit for a start. And really, I think the first thing I want to do is have a look at this interesting function here, right?
So we have to look at the equation. You are very correct. So can we first say, before we go into that function, in which sense is the equation actually very simple? If you look at the terms apart from the highlighted functions, they are all just variables.
Yeah.
Right, so it says minus x1 in the first line, and in the second line it says x1 minus x2. So we notice there are no rate constants. So what does it mean there's no rate constants? Well, there is a rate constant, but it's an implicit one. It's actually a rate constant set equal to 1. So 1 meaning it's an arbitrary value in this context, but something that helps us be simple mathematically. And so typically you would have multiplied this concentration with some rate constant that has some interpretation. So it's very much simplified, but it will help us understand the principal functions of these components better. Okay, then let's see the nonlinear function here. Maybe you first describe the individual components that are there, and then we try to think what it might possibly do.
Okay, so looking at a, so that's just a constant divided by km, another constant, plus ki, another constant, times by x2, so that's the variable x2 to the power n, and n is another constant.
Right.
So I can immediately see if x2 gets big, then this is going to be very small, and if x2 is small, this is going to be relatively large.
Right, so that's one of the tricks. Mathematically, implementing inhibition means we put things in the denominator, because then the regulatory molecule, in this case called x2, if it gets very large, it makes the whole term small, if it's a positive productive term. And if it gets small, it lets the productive term being large. So there's a way of sort of balancing things. And you can guess where the idea comes from. That is, if you have some downstream product accumulating very highly, and you have the idea that maybe this is getting too much, then an accumulation of a large amount of this downstream molecule might feed back into the beginning of the chain and telling the initial production rate to actually go down.
So let's have a look at that, shall we?
Let's look at it in a mathematical and in a graphical way to try to understand how it should work when in action.
So I'm going to make a new anonymous function called feedback inhibition FBINH. So we need to tell it what the variables, or including parameters in this case, are going to be. So we're going to have, well, let's just simplify x2 to x. We've also got a, we've got km, we've got ki, and we've got n. Okay.
Right, and closing the bracket means now you have defined all your variables. And as you can see, we have used it in the most general way, namely everything that we might potentially want to manipulate later, taken as a variable, and you should be familiar with what we have. Let me just take the occasion to mention that if you have any problem with the code that we are doing, or if you cannot reproduce what we are doing here, please use the chat functionality of the control panel to alert us. And if possible, if we can squeeze it in, we'll try to answer questions.
Okay, so we need to actually define the mathematical function now. So a divided by km plus ki times by x to the power n. Okay, that was a semicolon at the end.
So we have set our variables following the @ sign, and in the rear part of the equation we have the function defined. So we're not looking at the system of differential equation, just at this single function. And now how would we want to investigate this thing?
So it'd be nice to be able to plot this with the fplot function as a function of x. So in order to do that, we need to define another anonymous function that uses this one, but sets values for a, km, ki, and n.
Do you think we could call this x2 just to make it clear that we are dealing with only one of the two variables? I think it might be just clearer later on when we have it in the differential equation that this is what it actually means.
Sure. So let's define our new function, feedback inhibition 1, say, and that's going to be a function of just x2. And then we're going to call feedback inhibition. We don't know what x2 is, but then we're going to set specific numbers. We've got some parameters set up, decided here, so we'll use those ones, shall we?
Yes, let's put the values in.
1.1 and 1. Okay, and then we can use fplot to plot this function with some limits, of course,
Okay.
to 1. If I run this, I've entered the wrong. So there's an error, not enough input arguments, and I can see that the problem is I've said feedback inhibition rather than feedback inhibition 1. So feedback inhibition has 5 input arguments. Feedback inhibition 1 has only 1. So that's what I want. Let's try that again. That's better.
So we get a graph, and this is now open for our interpretation. What can we see from here? So what did you plot? It doesn't really say on the X. The X's are not labeled. What do we see here?
So this is a rate, I guess. On the y-axis, a rate.
On the vertical scale is a rate.
And on the x-axis is the amount of x2 in the system.
The amount of x2. So we can see how the rate of production of compound x1, our first metabolite, our first molecule of interest, depends on the amount of the regulatory molecule present, which is x2. So we have x2 on the horizontal axis, and if we just start at the beginning and we look at the case where there is no x2 present at all.
Yeah.
Right? So that's one way of starting to interpret the graph. And we have the case of x2 equals 0 displayed here. And what we can see nicely from that continuous graph that it starts at 10.
Yes.
So mathematically, does that make any sense? Why would it start at 10 and not at 20 or 0.1?
Well, if we go back to our equation, so A is 1, Km is 0.1, and this will be 0. So it'll be 1 divided by 0.1, which will be 10.
That's 10. So let me just repeat, make sure I understand. So you have said that if x2 is 0, this whole term will be 0.
Yeah.
So there will be nothing added to Km. So we are left with A divided by Km. And that's 1 divided by 1/10, and that gives us the 10 that we see in the plot.
Yeah.
If on the other hand we go to the right-hand side, we look at the concentration of x2 equal to 1, and it seems to have dropped to just the half of this initial amount, namely to 5. So I guess that also makes a lot of sense. Can we just look at our function again?
So in this case, x2 is 1 and ki is 0.1, so that is equal to 0.1. K plus km, which is 0.1, so that's 0.2. So 1 divided by 0.2.
So we have 1 divided by 0.2, and that just gives us the 5 that we can see here. Maybe, can you redo that plot and go much further than 1, say equal to 10, to say what we would now call a high concentration? So if we continue with this plot further up for even higher concentrations of regulatory molecule X2, you can see that this graph continues to fall down. Of course, it's 1 over the concentration, so we understand this hyperbolic decay here. And we're going to values below 1, and we might guess that for extremely large values, this will eventually approach 0.
Yeah.
Which I think is what we expect. If it goes to infinity, it would make sense that 1 over infinity is something like 0.
Yeah.
And we're probably not getting there in a biological sense, but we can understand the beginning, the dropping to half of the original value, and then the approach to 0 just by looking at these cases in the original function. Right, so an interesting feature of many of these regulatory feedbacks that it doesn't always follow this very simple case. So one way of modeling, of giving a certain flexibility to the model and being able to also approach shapes, experimentally measured shapes of this feedback that is maybe looking different, is to go and look at this exponent n.
So let's see what happens if we change n. So I'm just going to duplicate this, call this feedback inhibition 2, and I'm going to plot both of them. I'm going to make this a different color so we can see it, so let's make it red. And also I need to type hold on so they're both plot on the same plot. Should we change this to, should we say 2 to start with?
Make it 2. The important thing in these coefficients always is, as you might have recalled from the discussion of the Hill function that we did previously, is whether they are 1, smaller than 1, or larger than 1.
Okay.
So we have a case here that's larger than 1.
And we get a different shape of curve. So the blue curve is what we had before, and this red curve is what we have now. We can see that the inhibition starts off weaker. The rate of inhibition is weaker.
So for small concentrations of variable x2, the rate of production of x1 is higher.
Yes.
The red is above the blue one.
But once it gets past this sort of critical point, the rate is slower.
It's slower, and that means the rate for a given concentration, we have less rate or less production of x1 due to the stronger exponent, the exponent being larger than 1, with stronger inhibition for any value larger than 1 in x2.
I guess we can see the more extreme case if we say make this 100. Yeah, you're getting almost a switch.
So now we can, of course, get the idea, something that we have discussed before in the context of the Hill function, and we see it now sort of in an inverse fashion. Increasing the exponent lets us model switch-like behavior. So where for small concentrations there's no notable or very little variation in the rate, so it's actually not really affecting the rate, but once it gets past a critical point, it very steeply decays and goes to values near 0, so I mean switching off the reaction at all. And so we have an on-off model, and as you can guess, the value of 100 is exaggerated. We'll never see that in an experimental case. But we can see that switching type of functionality is approached when we see such a behavior experimentally.
Okay.
Okay, I think that's basically what we can say about this function. And we understand that depending on how we model it, whether n equal 1 or n larger than 1, we'll get hyperbolic decay or this kind of switching decay. And we can use that to say, for example, fit to modeling to experimental data.
So let's have a look at our model itself again, shall we?
Right, now we understand the critical part of it. The only nonlinear function in the model, we can actually look at the whole model, so two variables interacting together and displaying outcome that depends on the reactions between them.
So let's try and find the steady state. So we're assuming there is a steady state.
Assuming there is a steady state, we have to check whether or no. We hope we get it right because we have, as I said, modeled the open flow situation, which means that in principle it should allow for a balanced state, but we'll check in detail.
Okay, so let's set up our system. So we're going to have a new anonymous function. So we're going to do this algebraically first. We'll do it symbolically in a second. So a new system. So x is going to be a vector of two elements. So x1, x2. So we're going to let's just use our feedback inhibition one function as a shortcut. Remember we've got particular parameters set here, defined here. Or it doesn't really matter. We can change those later. But let's just start with feedback inhibition one to start with. And so x2 element x2 is going to be x with open brackets to close brackets. So the second element in x is what we're referring to by x2 now. And then that's going to be minus x1, which we refer to like that.
The degradation of the first species. Yeah.
So that defines the first equation here. And next we need to define the second one. So comma x1, capital X, sorry, minus x2. Okay?
Yeah, okay, so we got our first system, and the importance here to note is that this is sort of recurring back to a function that we previously defined, which is actually also good practice, but if you run that section individually, make sure you have actually executed the previous function to create this fb sub inh1, because otherwise it will be unknown.
So we can make it a little clearer if we just change this onto two lines. Okay, so our first, so we've got a function, an anonymous function, taking a vector of two elements, x, and returning a vector of two elements, which is the rate of change of x1 and x2. Okay? So we can then just solve this. So it's relatively easy in MATLAB. So the steady state is equal to, we use the fsolve function, pass it to the function, and pass it, are these initial conditions?
These are initial values. This is a guess. Essentially, that's fsolve will do a numerical estimate of the steady state, so it will iterate towards it, but in order to get started, it needs some initial guess. So what we do for an initial guess, it's basically arbitrary in the first place. If we are confident there's a unique steady state, we could take any values, but as we can't really be confident, we just have to take something and then see whether these are good values. So no. The only thing to keep in mind is that negative numbers wouldn't make a lot of sense because it would imply negative concentrations.
So let's run this and see what happens. Okay.
Running this section only, steady state comes out because you have not put a semicolon at the end. We can see it in the command window, and it says steady state has two values. The first and second, if we look at them closely, they're actually the same values, 2.7016, meaning the prediction is that the steady state for both will reach the same level.
So we can see that. So the steady state means when dx by dt equals 0. So that means dx2 by dt equals 0. So in this line, that's true. If that's the case, if x1 minus x2 is 0, then x1 must equal x2.
Right. So we can also understand that the fact that these two numbers are the same comes from the somewhat artificial assumptions that the rate constants of both processes are identical. Typically, they will not be identical, and if you have then proper realistic rate constants, then these numbers also will be different. And so we can see that this equality here is a bit of an artificial result mathematically because we have made this assumption, but it's also meaningful. Yep.
Okay, so we've done that algebraically. Should we have a go at doing that symbolically as well?
Just for practice, yeah, and also to have a bit of a wider repertoire. Symbolically is now the not numeric, but the sort of the explicit solution of the equation.
So we need to define the symbolic variables first using the sims command, so x1, x2. And then we need to define some symbolic functions. So f1 is equal to, it's going to be easiest now if we directly give it numbers. So say a is 1 as before, divided by km, which was 0.1, plus ki, which is also 0.1, times by x2. We could have to the power n, but we'll just set that as 1, as we've had initially, minus x1. And f2 is easier, that's just x1 minus x2. Okay? Just a different way of writing the same thing.
It's the same thing, but with numbers substituted in here for simplicity. So this a equal 1 or n equal 1, we have just put it in. Keep in mind that if you want to use those constants as constant and also potentially to change them, you would also have to define them symbolically and have sims also for the constants if enables that, and you could sort of treat it more generally. But for the moment, suffice it to have this simple way of writing the same equation in a symbolic way. And now let's find the solution of the steady states for this thing here.
Okay, we do that using the solve function. So the solve function, we need to set a vector of f1, f2, and they're both equal to 0 using the double equal sign because it's a logical equals.
That's a logical equal, right? And we have specified we want to have both equations or these functions that represent equations equal to 0, which is the same thing as above, but above we did it numerically. So also fsolve will then assume, we haven't really told it explicitly, solve means set it equal to 0.
So let's run this and see what happens. Okay, it's thinking. Okay, so it's thought and it's come up with an answer saying x1 is two numbers and x2 is 2 numbers. Okay, well let's save those. Let's save those as our, should we say x1 steady state and x2 steady state so we can have a look at them. If I run this again.
For those who don't know it, look this up, that if you want to have more than one output, then putting them into square brackets is the way to assign different names to them and then being able to look at them individually. Okay. And in this case, we can actually see the result rather than seeing that there is something. We can see what it is.
Okay, so they're
what actually is that?
So they're quite complicated numbers. There's 2 here, so there's the minus 41 to the power of a half divided by 2 minus a half, and similarly for the others. Actually,
what does it mean? We get 4 results, right? We get 2 x1 steady states and we get 2 x2 steady state values.
So this is a steady state value for x1, the first one and the second one, the steady state value for x2, the first one and the second one. So we want this number and this number are a pair, and this number and this number are a pair.
Yeah, is that clear to everybody? So that if we get pairs in each case, it means that we have found 2 steady states. And the first in x1 corresponds to the first in x2. They form a pair that tells us what the steady state of the system is. Similarly, the second number in x1 and the second number in x2 form the second pair. And in principle, it could be more than this. There could be 3, 4, 5 steady states, and in which case we would get 4 numbers for x1, 4 numbers for x2, and so on.
So if you run those, so these are the right pairs. So we've now got 2 negative numbers as a pair and 2 positive numbers as a pair. But again, they're not very useful. We need to we don't really want 41 to the half, but we can use the eval function to make MATLAB turn it into a decimal number rather than an expression.
All right, let's just evaluate it. This is eval and evaluated,
Okay,
and
that's
we
better.
do get two numbers for each pair.
So we get the same pair as we had before, the 2.7.
We get the 2.7016.
But we also get a minus 3.7016.
Yeah, and this means this set of equation taken generally has more than one steady state. So actually doing it symbolically and sort of not depending on doing a right guess in the first place, we get actually two numbers, and means there's two steady states in the whole system.
Yeah.
Of course, looking at these numbers, minus 3.7, we'll discard the negative one because we don't want to look at negative concentrations. That doesn't make a lot of sense, but mathematically it's there, and it's the symbolic way to tell us what its value is. So let me just quickly repeat this. The second one was the one that we were interested in, so it doesn't move too fast. You have grouped the second value for x1 and the second value for x2 together using these parentheses to put them into a small array of two numbers. And the eval function around it just tells MATLAB to not give us some expression, but give us the actual numerical value.
Yes.
And as mentioned in the beginning, you have all this all worked out with comments according to what is happening in that downloadable file under materials.
Okay. So we know what the steady states are, but now we should be nice to see the actual simulation of the model, correct?
Right. An important feature of the model, and especially when we look about signaling cascades, where this original example was taken from, also in gene expression regulation, is not so much what is the actual steady state, but how is the transient behavior towards that steady state? How is it reached, say, following, say, agonist stimulation, if you look into signaling networks, or say endocrine hormone stimulation and such things. And then the transient behavior, how the model responds to a certain stimulation, also to a perturbation, thinking maybe of drug discovery. If we want to know that transient response, we can't really do much with the steady state, but we need to make a simulation of how that steady state is approached. So let's do that.
Okay, so we could, and in lots of cases it'd be best to make a new file for a function for the ODE model. But because our system is actually quite small and quite simple, we can just do this using anonymous functions again. So we're going to use ODE45 to simulate our model, and that expects a function, let's say f, that needs to return two values, t and dx, which is a column vector. Sorry, it needs to take t and x, sorry, my mistake, and it will return dx, so dx by dt. Okay, so let me clarify again. We're going to use the ODE45 function to simulate our ODE model, which requires a function taking the current time, which we're not interested in, but ODE45 expects it, and x, which is going to be a column vector of our concentrations x1 and x2. That's going to return our dx by dt, so dx1 by dt and dx2 by dt as a column vector. So let's write that. So f is equal to at t x, and then we've written our feedback inhibition function before, so let's use that one. So that second value is going to be x2, and then the weight constants we have here, so this is the right order, so it's going to be 1, 0.1, we're told to use 0 to start with, and then n is 1. Okay, so there's no inhibition to start with. Okay?
Okay, so I have a simulation of just the pathway, so to speak, the linear pathway without any further regulatory features.
that's going to be, subtracted from that is going to be the value of x1. Okay, so that's the first differential equation written out in MATLAB code. And then we need a column vector, so a semicolon. I'm going to make this on a second line just for clarity. We have x1 minus x2 as our second differential equation. So dx2 by dt is equal to x1 minus x2. Okay?
Right, so yet another implementation of the two rate equations recurring to that feedback inhibition function that we defined above. So we'll need to have it defined before. And importantly, with the notation that uses t comma x, or using two variables, because that's the way that the simulation, the numerical integration is actually requiring it.
So then we can simulate the model using ODE45. So that will return the time and the x values. ODE45, give it the function f, give it the time, so the minimum time and the maximum time, and then give it some initial conditions. And then all we want to do is plot the output. So let's just use plot. Okay.
Time will be on the horizontal axis, and the concentrations of both variables will be displayed if we do that. And so the remark that I would have, this is slightly different from what we teach in the materials, where we have a script that executes based on a model, and the model is actually written separately in a function file. So you can see this is an alternative way doing the same thing using ODE45.
If your function is small enough to be written on a few lines as an anonymous function.
Right, so you wouldn't necessarily do that for very complex biologically realistic models, but for this very simple thing, we have it in a very manageable way in just a few lines here.
MATLAB treats anonymous functions and functions in separate function files basically the same. I think completely the same as far as I'm aware.
Yes, it's very similar treatment, and you wouldn't notice any difference. So the convenience here being that we don't have to switch between different files, but we have them actually one above each other, and we can sort of interact directly. Also conveniently, we have in the use of the anonymous function an accessibility to the parameters, which just lets us manipulate things easily. So to recapitulate, our parameter a is equal to 1, our km is equal to 0.1, what we looked at before. And we are first doing a simulation without any feedback inhibition. So ki is equal to 0. And we have an n equal to 1, so we have this type of decay that we recall from, continuous decay that we recall from previous discussion.
All right, let's see what happens. Okay. So we're plotting both x1 and x2 values here. I'm not sure which is which. Perhaps we can figure it out.
How can we figure it out best? An easy way would be maybe to go into the graphical user interface. You can, of course, also make an annotation, but it shows that the blue one is the first variable.
So x1.
x1, and the red one is the second variable, x2. So let's take a quick look. What does it show us? Their increase does not follow the exact same route, but they're slightly different. Notably, the second one follows. The first one rises first, and the second follows. So both start at small values, but it's the first one that rises first because of its, of course, being the first in line. And having no feedback, the second will just be produced when the first one rises to a certain concentration, proportional to that concentration. The value 10 sounds somehow familiar to us, because we remember that was the maximum value that the function took for no concentration of x2 present at all. So what we can see here is not that the concentration is 0, but that the concentration reaches this high value because there is no feedback. So now it's not the concentration 0, now it's the Ki, which is 0. Yeah. And that leads to both leveling off at the value of 10. We understand why they are both the same. And the delay also understandable because 2 follows 1 in the succession of the chain. So why don't we include some feedback inhibition?
Okay.
That's what we wanted to look in the first place.
Close that.
So let's include that and have a simulation with small value for Ki, that's 0.1. And if we repeat the simulation, this is what we get. Remember, blue is x1. So it starts from the same original value, rises, but now rather than approaching 10, goes to a maximum at 3.5, and then decreases. So it's a novel behavior.
And of course, that level, that steady state, is the 2.7 we had before.
Actually, now we're getting this 2.7 steady state that we calculated in the presence of feedback inhibition. And we're not surprised that it reaches this, but we also now see the transient and especially this overshooting, which is a very typical way of reacting. If you have feedback inhibition downstream backwards, then overshoot is a very clear indicator, or one of the signals that tells you maybe we'll look for feedback inhibition if we see that in data. Right. So could we overlay this with the previous case of no inhibition?
Yes. Hold on. I'll just copy materials again.
So if we have a copy of the code, we return this Ki value to the original 0, and keeping the previous experiment, this simulation will lead to the original curve, namely no inhibition. And what we can see is that inhibition actually means the steady states have been decreased. So the blue and purple curve are now our original uninhibited or no feedback network. And we can see they rise to this high level 10.
The yellow and purple.
The yellow and purple, yellow being x1. And if we compare the feedback inhibited case or the regulated case reaching a much lower level, you know the value is around 2.7 in this case with these constants, and it shows that at some point during the initial rise, the fact that variable x2 is being produced starts to exert its feedback inhibition and keeps the blue curve from following the yellow curve exactly. It gets less and less and goes through the aforementioned maximum, then leveling off to that lower value. So we can see that the overshoot phenomenon also leads to a total decreased steady state values for both of these variables. So that would be sort of our prediction from this case, and essentially sums up our discussion of this model. One thing that we could do, if we just take this whole thing away, is to look at what happens for the extreme case of some much higher exponent in n.
Okay,
so if we look at n equal
10.
2 or 10, I don't know, what shall we do, something extreme? So make it much larger and put it equal to 10. The reason we are doing this is to alert you to a thing that's long been known in engineering and the theory of feedback systems, where feedbacks are actually used to stabilize processes at a desired value. So if that level 2.7 was not 2.7, if the steady state is here, if that's a desired state, feedback inhibition is the way to control a system to that state. And biologically, that works in a similar way. And so the engineers have always been warning us that if you make this feedback too steppy or too sharp, if you like, and we have this implemented by n equal to 10, a large exponent in the feedback inhibition function, then what you'll see is an oscillatory approach to the final state. Oscillatory meaning that the former overshoot will be followed by an undershoot, another overshoot, undershoot, and hopefully these oscillations are damped, so we'll still get there, but there might be considerable wiggling before we end up there.
Let's make that even more extreme and see what happens. It's a very considerable
making
wiggling.
it 100, and we remember what it meant, right? Because we showed the case of n equal 100 before. It's this very extreme step function that we used. And if we do the feedback with a real switch on step function, what we can see that not only do we get this oscillatory approach, this damped oscillation towards the steady state, but also the waveform is now impacted. In contrast to the previous more sine wavy, smooth-like approach, we're now having something that's more edgy, or looking maybe sawtooth-like, whatever nomenclature you find for this. And it shows that this is now an extreme case of a nonlinear feedback regulation, leading to non-sinusoidal or asymmetric waveforms in the response to a certain input. For those interested in those things of feedback regulation and also some engineering principles, there are cases, and also what the Goodwin model was originally made for is to actually describe oscillatory behavior. So where the oscillation is not damped as we have it here, but an oscillatory behavior that is maintained because that has been observed experimentally. You can see one way of getting to this point if you look at the script that you can download from our materials page, worked out by the use of delayed feedback. So that's something that we don't go into detail now, but delays in the feedback considerably enhance the probability of seeing oscillatory solutions. And for those who now ask what is that actually useful for in biology, we don't explain, but we'll hint at one point this principle, and specifically based on the Goodwin model, has been used in a paper that's actually has quite some age. It's been published in 1999, followed by many, many more refined models where the circadian rhythm in eukaryotes has been modeled using this Goodwin approach. So you have the citation here if you want to look it up in the Biophys journal, and look also for papers that cite this one. The circadian rhythm can be explained in a first approximation just using this feedback inhibition functionality. And if we go to the references, at some point it shows that actually this is all based on just this Goodwin model that we have discussed here. And you can see if you look up this paper that this is a very accepted framework now of explaining known experimental features by a more detailed mechanistic model, where actually the X1 and X2 are replaced by proper known proteins that regulate the the autonomous variations in the circadian rhythm in eukaryotes. Among others, this is also happening in the human.